Hey guys, welcome to Cosmic Solutions, Dr. Priyanka's Biology. This is a channel where we discuss everything from biology to biotechnology. So today we are going to deal with our NCERT chapter, Principles of Inheritance and Variance. This topic is very important from your board exams as well as NEET exams point of view, because the laws of inheritance, that is the major thing that we are going to focus here, and these things are like repeatedly asked for your board exams and also your NEET exams. The application part of you have to apply this laws of inheritance to solve some problems. So if you understand the entire concept, it will be very easy for you to solve the problems with you in your NEET exams. So let's get started with inheritance, principles of inheritance and variance. So what is meant by inheritance? Suppose there is some property, some piece of land, some mansion that belong to your grandfather. So from your grandfather it will come to your father, and from father it will, from your father it will be passed down to you. So basically it's a sequential passing on of the characters, or passing on down of the property. So that is what is called as the ancestral property, right? Same way, in genetics or in biology, we talk about the inheritance of genes. The certain characters are passed down from parents to the child. That is what is inheritance of genes. So, for example, when a child is born, his aunties and all, they come and they say, okay, the eyes are exactly like the mother, the nose is like the father, okay, the color is like father. So all these are genetic characters that have come down from the parent to the kid. These aunties are just trying to figure out like with whom the resemblance is more. Okay, so this is what is inheritance. Okay, so this particular study was initiated by a person or an Austrian monk called as Gregor Mendel. So Gregor Mendel was an Austrian monk. He was when he was working at Saint Thomas Monastery. Okay, he had the environment of laboratory. He had the facility of laboratory, which uh, made him interested in research and teaching. So he started working on certain pea plants. Okay, he started carrying out some crossbreeding or hybridization experiments in the pea plants, and then he was working on those fields. He started noticing the characters in these plants, okay, how this variation is occurring. So in this way he started working on all these characters, and together he put forward certain laws which are today called as law of segregation and law of independent assortment. So Gregor Mendel is called as the father of genetics. So after his death, his work got recognition, because in olden days there were not systems of like today we have internet, we have computer systems. Okay, the communication has become very easy. If somebody is working on something in India, the person in USA will come to know because this work is getting published, right? So in olden days, these facilities were not there. So after many decades of his death, his work got recognition, and all the work that he did, it formed the basis of genetics. So that's why he is called as the father of genetics. So what he did was, he was working on garden peas. So almost for 7 years he worked, and he proposed the law of inheritance. So he was the very first one to combine mathematics and biology. He used mathematics to solve the problems in biology. So funny thing is, like many people who are studying, they do not like mathematics much, right? But he was a person, he combined both the studies. Again, contrasting characters. Just a joke. Okay. So there were certain special things about his work, that he had very large sampling data. Okay, the sampling size was very vast. That means he used the entire fields to study this thing. This had led to this has added to the credibility of the data that he had collected. For example, now you guys must be now familiar with the chemistry labs that you are working with. You have to perform the test 3 times, or you have certain tests in which you have to do the CT or the confirmatory test. Then only you can report to the examiner or your teacher. So even when we are working in lab, certain experiments, whatever we are doing, we always do them as triplets or thrice because we want to be sure whatever work we have done, whatever data we have carried out, or whatever experiment we have done is proper. It is not error prone. So this particular concept was used there. That there was large sampling size. Okay. So all these things it gave the confirm after working on so many plants. Okay. This all these things he saw certain similar results which led to the penning down of the laws of inheritance. So let's get started with Mendel's law of inheritance. So here he studied the opposite traits. Okay, he worked on different opposite characters. For example, tall and dwarf plants. So, or whether the seed is constricted or full. Okay, such contrasting characters were studied. So, because of this, it helped him to put down the rules, okay, which govern the inheritance, which further helped the scientists to work further and establish proper laws. So what did Mendel do? He carried out artificial pollination or cross-pollination. Okay, usually plants do self-pollination, but because he wanted to study the contrasting characters, so what he did was he started doing the cross-pollination or artificially he was pollinating. So he had selected 14 true breeding pea plants. Okay. So we shall see what is true breeding later. So he used this 14 different plants. So that means 14 meaning they had 7 pairs. Okay. So they had everything same except for one particular character. For example, when he was working on seeds. Okay. So everything about the plant was the same, but the form of the seed, whether it was grey and round or wrinkled and white. Then next, the cotyledons, the color, yellow and green. The flower color, white and purple or violet. The pod, the shape of the pod and the color, that is full and constricted, yellow and green. Then stem, whether the pods are placed axially or on terminals. The size, whether the plant is tall or dwarf. So in this way, he had a full set of 14 true breeding plants. Okay, with that means the 7 pairs with contrasting characters. So what he did was, so what did Gregor Mendel do? He cut off the stamens. So if you see this plant over here, if you see this picture, the stamens, they are basically made up of the anther and the filament, indicating the male parts of the flower. So what he did was he removed this, or he cut off stamens from the purple plant, and second one was he had taken a white plant. So what he did was he took the sperm bearing pollens from the stamens of this white plant and he transferred them to the carpel or the female part, the stigma and style of the male plant, sorry, of the violet plant. So after doing this, he entrapped this flower in a bag. Okay. So what he did was to allow any prevent any sort of cross pollination, as well as the removal of stamens was to prevent the self pollination. So after this, after successful pollination, the plant matured and it led to formation of matured pods. So the seeds from the pods, they were put in the soil and they were grown. So it was seen that all the plants produce the purple flowers. So he started noticing, okay, what I had done was I had taken 2 different plants, right? So that one was white, one was purple. So now when I had sowed the plants, sowed the seeds, how come everything turned out wrong? Purple and not white. So these were some of the questions he tried to answer. So before we move on, let us try to understand or figure out two different terms, that is a genotype and a phenotype. So genotype refers to the sequence of DNA. So it is two alleles of organisms that have inherited a particular gene. So we have this genotypes could be of homozygous, recessive, homozygous, dominant and heterozygous. So when we are saying homozygous recessive, so it is indicated by the letters, that is the small pp. This is just an example. Homozygous dominant, that means both the characters, both the alleles, they will be dominant, that is capital P, and heterozygous, that is the two different. So we have a combination of capital P and small p. So next is the phenotype. So what is phenotype? Is something the observable characters, so characters that we can observe. Okay, so that is what is your phenotype. So they normally result in expression of one or more genes. So phenotype, for example, we have the brown color hair, okay, the eye color. Okay, these are the some of the phenotypic characters. Then when it comes to blood group, we have A, B, AB and O type. That is also a characteristic of a phenotype. So let us try to understand the Mendel's law of inheritance. So here let us try to understand certain terms, that is the dominant and recessive. Obviously we know dominant indicated by a capital T and recessive indicated by a small p. Okay. So now let's try to understand one term, that is the monohybrid cross. So here we are going to deal with only one type of gene. So a monohybrid cross is a genetic experiment involving two individuals with homozygous genotypes. Okay. And these individuals have completely dominant or completely recessive alleles for a single trait. Okay. So that is what is called as your monohybrid cross. So these crosses are normally used for studying the inheritance of a single gene. Here if you see, we have taken a tall plant with a genotype TT, capital TT, and a dwarf plant with small tt. So what we have indicated in these rounds are the alleles. So when we carry out a cross between such a pair, so we will have, so here capital T is dominant. So phenotype will be like the T plant. So capital letter. So we have a capital. Okay, then again we have capital T small t. Then again we have capital T small t. Then again yet another time we have capital T small t. Now here if you see, all are a combination of capital T and small t. That means the dominant character is going to show the phenotype. So we have capital T small t. So that is the F1 generation or the first filial generation. So all the plants, they are tall phenotypically. So the genotypically they look the same and phenotypically also they are looking the same. See the next thing, that is so now here what we have done is we have crossed the products from the first F1 generation, that is capital T small t, capital T small. So they both are tall plants, right? Both are tall. Okay. Now once we cross them, capital T, that means it's a tall plant. Small t, capital T, again it's a tall plant. Again we have small t and capital T. This is also going to be a tall plant. and small t, it will be a dwarf plant because both are recessive. So if you see here, 3 are dominant and 1 is recessive. So and this is, if you see, this is a phenotypes, tall and dwarf. The phenotypic ratio would be 3:1. That is, we have 3 tall and we have 1 dwarf plant. So the phenotypic ratio is 3:1. And if you see the genotypic type, so capital T, capital T is of one type. So that small t, capital T, we have 2. And again small t, we have 1. So the genotypic ratio is 1:2:1. So while he was working on this, so he saw that, like, if you see here, we were talking about there were no dwarf plants. The parents were not dwarf, but somehow, okay, one of the progeny in the F2 generation or the second generation became dwarf. So there was something that got passed down, right? So this small t, which was there in the first generation, okay, that got passed down. So that means there was something that was getting passed down. So Gregor Mendel did not know the technical term. So he called it as factors. Okay. And the genes that code for a pair of contrasting traits, they are called as alleles. So they are slightly different form of the same gene. So we have a capital T, small t. So these both are alleles for the tallness or the dwarfness of the plant, height of the plant. So after understanding this, okay, he had put down certain terms. So he came to know the characters are controlled by certain units called as the factors. These factors, they will always occur in pair, okay, and in dissimilar pairs. In dissimilar pairs, one member is going to dominate the other member. So we have a dominant and the recessive alleles. Alright. So let's move on to a small part, that is a Punnett square. So let us see what is a Punnett square. So whatever we have studied so far, okay, so there was a geneticist called as Reginald Punnett. So he proposed something called as a Punnett square. So this Punnett square is a method, or it's a pictorial or graphical representation which will help to calculate the probability of all the possible genotypes of the offspring. So if you see this square what I have here is nothing but a Punnett square. So wherein we can study the pollination or the crossing of the two different characteristics of plants. And then based on this we can study further. Then let's understand one term called as a test cross. Now this test cross is done specially when we are unaware or when we do not know one of the genotypes of one of the parents. So crossing an individual with dominant genotype or phenotype with another organism exhibiting the recessive genotype. So basically we are going to cross a dominant genotype or recessive genotype. Okay, sorry. We are going to cross. So I'll explain you here. So if you have one goat whose whose phenotype is known, this should have been genotype here. I made a mistake. Whether it is BB or capital B, small b, we do not know. And another one, this mistake, please, I have to rectify this. So another one is the homozygous recessive. Okay, so that means it is going to have this BB. When they are being mated or when the crossing is done, what will happen is, now here we have two possibilities, whether it is capital B or small b, we do not know, right? So let us consider both the cases. That is, suppose if the unknown genotype, again missing, genotype is capital B, capital B. So what is done is the capital B, the alleles are put here and the alleles of this recessive one. So we know it is a recessive one. They are put over here. So crossing of this, it will give rise to capital B, capital B, capital B, small b, capital B, small b, capital B, small b. So all will have the small b, capital B composition. So all are going to be brown. So this is when the genotype is dominant. So when the genotype, okay, is homozygous, heterozygous, okay. So we have here capital B, small b, and we are going to pair it with the recessive ones. We have capital B, small b, okay, small b, capital B, small b, and small b, small b, right? So if you see, we have two capital B small b, two capital B small b, and two small b small b. That means two will be brown and two will be white. So that means the ratio will be 1:1. So that is the difference between the test cross of the dominant types and the recessive types. So this you have to keep in mind. Right? So here is another term, that is something called as back cross. Okay. So back cross is a type of mating, okay, mating of hybrid organism, offsprings of genetically unlike parents with one of its parents or with an organism genetically similar to the parents. So back cross is done for studying the studies for isolation, okay, of certain characteristics in a group of related animals and plants. That is what is a back cross. So now let's come to the last part for today's session, that is the law of segregation. So the term segregation meaning to separate out. Okay. So law of segregation, what does it state? Okay, let us try to understand. Right? So this law, when we talk about the law of segregation, okay. So here it says that, okay, the two pair of alleles, they are going to separate or segregate at the time of gamete formation or meiosis, okay, and such that only one allele is present in each of the gametes. So this is what we have studied in our normal biology. So these homozygous plants are going to produce all gametes same, whereas the heterozygous, they are going to produce the two different kinds, okay, which will have the equal proportions. Now this law is based on certain concepts. That is, first thing is that a gene, they are going to exist in multiple forms. Second one, when gametes are produced by meiosis, the alleles pair separately, leaving a single allele in each of the plants. Fine. Then each organism is going to inherit two sets of alleles of a particular trait. Then next is two alleles of a pair, they are different. That is a heterozygous. And one allele is dominant than the other one. So that is, one is dominant and second one is a recessive one. So this is the first law, that is the law of segregation. So in coming session, we will be discussing about yet another topic, that is the incomplete dominance. So till then, keep studying and keep smiling, and don't forget to like our channel and subscribe. Thank you.